I'm Susan Brawley. I have spent a lot of my life since I was about 12 years old tremendously interested in the macrophytic algae, which are commonly called seaweeds. Our local Charlotte, North Carolina Children's Museum had a trip to the North Carolina coast when I was 12. And someone who had been the associate director at Duke Marine Lab actually led the field trip. And I can remember every minute of it. I even used some of the exercises we did on that trip at the University of Maine in my marine biology classes when I came here and started teaching marine biology.
My name is Jessie Muhlin. I'm a professor of marine biology and the chair of the Corning School of Ocean Studies at Maine Maritime Academy. And I'm a phycologist, someone who studies algae, and that includes seaweeds. I've been at Maine Maritime Academy, this is my 17th year, and I love teaching students about seaweeds and algae in general, and do research on the side when I'm not teaching in the classroom. I'd say hands down the food that the seaweed product I use the most in my kitchen is called Triple Flake by Maine Coast Sea Vegetables. It's a dried and flaked seaweed that has dulse, which is a red seaweed, sea lettuce, which is a green seaweed, and nori, which is a red seaweed.
Life on this planet would not be the way it is in any way, shape, or form without cyanobacteria. So perhaps as long ago as 3.5 billion years, and certainly by 2 billion years, the cyanobacteria were producing a lot of oxygen. So photosynthesis arose in the cyanobacteria, and one of the byproducts of photosynthesis is oxygen. So that turned out to be very important to our atmosphere.
We wouldn't be here without algae, but full stop. And I think most people don't really sense the gravity of that statement, but it's really true. And so 2 billion years ago, they started to capture light energy and create their own food from that light energy. And as a byproduct of photosynthesis, they also evolved oxygen. And so for a really long time, like billions of years, millions of years, that oxygen started to accumulate in the atmosphere and fundamentally change the geochemistry on Earth.
Almost all of the organisms that we meet as we walk around during the day have to have oxygen.
So seaweed has evolved many different ways and is continuing to evolve. And so from the simple cyanobacterium, there has always been a diversification of cyanobacteria. So there's a lot of different types of cyanobacteria. They inhabit both the marine environment and freshwater environments. They're found so many different places, also in terrestrial environments. But from them, they diversified through a process called primary endosymbiosis, where something that was not photosynthetic, called a protist, a eukaryotic protist, ate a cyanobacterium and kept it inside of its body and then incorporated it as an organelle or a plastid. And that gave rise to a whole host of different algal lineages. And within the seaweed specifically, it gave rise to something called the glaucophytes, the red algae and the green algae. And in the marine environment, we have red algae, green algae, and brown algae. And the seaweeds are represented by those three lineages, that their great ancestor was a cyanobacterial cell. All sorts of different ways. So the algae and seaweeds can have different kind of characteristics that you can see with your naked eye. They have different characteristics you might see needing a microscope, and it could be a really coarse microscope called a dissecting microscope, or a compound-like microscope, or something even more sophisticated in terms of the magnification. Nowadays, people use molecular techniques, looking at genes either found in the nucleus or in the mitochondria or in the chloroplast to differentiate and identify different groups of algae. And some of those morphological characteristics that you can see have to do with what the cells look like, what the overall structure looks like, what their biochemistry is, or what their pigmentation is. Yeah, so pigments are really interesting molecules in which they have a number of different functions for photosynthetic organisms. And for the seaweeds themselves, the fundamental cellular machinery for photosynthesis is a molecule called chlorophyll A. And chlorophyll A is that bright grassy green, kind of emerald green color that's characteristic for all photosynthetic algae. All photosynthetic algae have chlorophyll, but they could be masked by other pigments. So the brown algae and the red algae have what are called accessory pigments that kind of stud or go on top of those chlorophyll molecules, and they are able to extract different wavelengths of light that the chlorophyll molecule is not able to harvest, and they transfer it to the reaction center, or the cluster of chlorophyll that actually does the photosynthetic process. So it has to often do with depth, but not always having to do with depth. How light behaves in water is different than how light behaves in air. And as soon as you enter the water, there's something called light attenuation. So different wavelengths of light penetrate to different depths of the water. And so very quickly we lose the red wavelengths of light, the orange, the yellow, and the green, indigo, and violet wavelengths of light. And so those accessory pigments that the brown algae and the red algae have are able to harvest those wavelengths of light that chlorophyll is not able to access and harvest. And they can live deeper in the water column than, let's say, the green algae. So you typically find green algae higher up on an intertidal zone, somewhere in between where the tides are. And then typically, but not always, there's then a brown algal zone, a brown seaweed zone. And then the reds, with their photosynthetic accessory pigments, are able to inhabit deeper in the water column. And then also the kelps are also kind of subtidal or very low intertidal.
So it's just gotten more complicated. We find green algae deep, even though most green algae are shallow. We find brown algae from shallow water down to a couple 100 feet in terms of large kelp beds that can occur at that depth. And the reds are everywhere. So this is my laboratory at the University of Maine, and I have a microscope room that we'll probably see later, and there are offices where people can do their paperwork and thinking. But the laboratory is equipped to allow for a wide variety of activities. And so in this lab, things from marine ecology to microbiology to cell biology to understanding how to get the genome of a red alga called Porphyra umbilicalis. They've all been done here.
It's a pleasure to know that people are learning about seaweeds and algae in general. I think it's one of the most important groups of living organisms that people don't really get a chance to really learn about. They're typically stereotyped as being slimy and gross and not important, and they really are so important. So I feel like the more you learn about them, the cooler they get. And so I really appreciate you having an open mind to enjoying and learning more about seaweeds and algae in general.
You should look for algae everywhere. And so when you study algae, when you learn about algae, you're not just learning about some little group. You are learning about organisms that have made the earth what it is. The algae are basically so diverse in terms of their evolutionary organisms after the last eukaryotic common ancestor diversified. But if you are interested in the algae, you can study anything.